Hello, this is Dr. Bradley Alicea, and I'm going to be giving an update on the activities for DevoWorm over the year 2024. So that's from November of 2023 to November of 2024. So this is the 10th year anniversary of DevoWorm. We've been doing computational developmental biology for a decade. So you can see in the background image, we have this timeline with different types of projects on it. And I was pleasantly surprised going through all of the things we've done over the past 10 years, that we've done such a diversity of things, and how those approaches have been incorporated in our talks and papers and meeting activities. And so there's a link to our YouTube channel where I've put an hour and a half long video on 10 years of computational developmental biology. So DevoWorm started in April of 2014. So it actually was initiated by a GitHub issue, and that was on the OpenWorm Foundation's organizational GitHub, issue number 179. So you can see that Stephen Larson put this issue up. He basically put some goals on here. Basically, find a data source that allows us to find, the lineage tree for C. elegans, which is well known and published, and bringing that into a computational environment. So we did this with our first paper. We worked on this. We produced an RDF representation of this, and it was a good starting point. But we didn't directly solve this problem because we didn't incorporate it into PyOpenWorm. However, we did do a lot of other things after that, and so our first meeting was in April 11th of 2014. The attendees were Stephen Larson, myself, and Dr. Richard Gordon, and we met in Google Hangouts, which doesn't exist anymore. But we talked about how to sort of get started, and we started that first paper, which is on the archive, and then we continued on with many other projects over the years. So every year since 2017, we've been involved with Google Summer of Code, or GSOC. And this year we sponsored two students. We sponsored Mehul Arora, who did this project called HyperDevoGraph, which is creating hypergraphs for the C. elegans embryo from the lineage tree data, and Paki Bonchalia, who did a project on growing developmental graph neural networks with neural developmental programs. And so both students contributed to the DevoGraph repository of DevoLearn at this GitHub address, and they contributed both of their projects as directories in this repository. So if you're interested in contributing in an open source fashion, please check this repository out. So the first part of Paki's project was the application of the Kepler mapper or K-mapper algorithm to the C. elegans lineage tree. And so this is on the left, sort of a visualization of the lineage tree in terms of topological data analysis, which allows us to look at sort of the shape of the data. So the data has a certain shape. We want to characterize those shapes and visualize things so that we can get a better handle on the data. This was visualized in a Mathematica applet specifically for this purpose, and it looks kind of like a UMAP. Although it's not, it has a bit more information in it and different type of information. So this is a fork of Luca Simi's TDA mapper in Python. So that repo was forked by Paki, and we were working from that fork. Topological data analysis was conducted on the C. elegans lineage tree data. It allowed us to preserve the cell nomenclature, the line of descent and positional data, and to characterize how those sort of fall in terms of shape. And then this contributes to a characterization of the underlying shape of the data. So one step in building graphs is to characterize kind of the connectivity and the sets of relationships between the nodes, which represent the cells of a certain nomenclature, a certain line of descent in that lineage tree, and a certain position in the embryo. The other thing that Paki did was neural developmental programs. So this was forked from Elias Najaro's GitHub project, which is this NDP project. And this wasn't made specifically for what we were trying to do, but we tried to work around it the best we could and modify the neural developmental programs. Basically, it allows you to grow a network from a single cell to multiple cells. And they use this for neural networks, but we can use this for our own uh, uh, embryo networks and specifically tying it to the lineage tree and to the spatial position of cells. So this model was trained with C. elegans embryo data for the purposes of GSOC. The network grows as the cell divides. You can see on the left you have this visualization of cells expanding and then the connections between them. And the way that these graphs are plotted out is that these are kind of force-directed graph projections, so they're not always going to match what it looks like in the embryo. In any case, we have different connectivity criterion and clique membership. So clique membership is where you have a bunch of nodes that are tightly connected together, fully connected. And usually those match up to the lineage tree. So this means that these cells have a common set of descendants and have a common sort of a common part of the embryo. So there's some spatial distance between them, but they're sort of clustered in the same part of the embryo. And so Pocky's project was this graph neural network, neural developmental program hybrid model. And I think we made a lot of progress on it this summer. We're trying to work on it for next summer. And so if you're interested in participating in GSOC for next summer, let us know. We also had 3 lectures during 2024. The first being this Mathematics of DevoWorm presentation, which goes through all of the mathematical models and developmental computational representations we use in DevoWorm. So this is where we have different models for connectomes, for embryos, for modeling behavior, and all of these things, and kind of it's an exposition of all of that work. So this was presented at the Society for Mathematical Biology's Cell and Developmental Biology Festival. This was held virtually, and you can find the recording of this talk here on our YouTube channel. So then we also had 2 other talks. It was basically an extension of the work we've been doing in our meetings. The first is called Dimensions of Morphogenesis, which are different models for morphogenesis covering 1-dimensional, 2-dimensional, and higher dimensional models of morphogenesis. That can be found on our YouTube channel as well. And then a 10,000 meter view of DevoLearn. So DevoLearn is the software package that we've been building over the last 5 or so years, putting in all sorts of things, pre-trained models, graph neural networks, different types of models like segment anything and so forth. So this is, kind of coming together. We've had a lot of people work on this, and that can be found on our YouTube channel as well. We've also done a number of papers this year. The first paper was this molecular basis of differentiation wave activity in embryogenesis. This was myself, Sarosh Bastani, Natalie Gordon, Richard Gordon, and Susan Crawford Young, and this was published in Biosystems a couple months ago. And the idea behind this paper is we're trying to identify signatures of these things called differentiation waves. So differentiation waves, it's a sort of reinterpretation of all the different types of waves that are found in development. We have all sorts of waves such as calcium waves or signaling waves or other types of waves that go through electrical activity that go through populations of cells. So there are these different waves that happen in tissue and cell populations that drive changes in cell type and differentiation. So you have some array of cells in embryogenesis. There's a wave that goes through them, and then there's a differentiation mechanism that gets triggered in these cells, and they differentiate behind the wave. So this is, just a way to reinterpret the literature, but also a way to describe, what happens when you have collective differentiation, when you have differentiation over a whole bunch of cells, and you end up with this sort of tissue morphogenesis. And so the other aspect of this paper is we used 4 model organisms. We used secondary genetic and proteomic data to look at 4 different systems. C. elegans, of course. That's where the bulk of the work was conducted. Drosophila melanogaster mouse and yeast or Saccharomyces cerevisiae. So we got proteomic data for all of them and genomic data for C. elegans. Then we had these models that we used to understand the interactions between genes and C. elegans and how they relate to protein expression. And so we could then map that to sort of a model of how these things might set up differentiation waves and act in development. So that required us to do some literature search for candidate genes and candidate proteins, and to get some secondary data sets on gene expression in early and late embryogenesis. So once we had the gene expression data, we could build these interaction models, which are not genetic regulatory networks, but they're fully connected and sequential models that allow us to sort of understand the relationship between candidate genes and proteins and their contribution to things in development. And so this is another example of some of the work we did here. We had a bunch of genes and looking at their variation of their expression and development and their expression level, and building a surface that describes how those genes are related to proteins, what proteins are most active in development, which proteins are least active in development, and what that surface looks like. So you can see that there are some genes that are very interesting from this plot and some that are less interesting, but maybe also important in some of these cascades involving the propagation of these waves and then what happens after the cells get that signal and start to differentiate. So we have our gene expression space on the left, and on the right we have an example from Drosophila, the imaginal disc in Drosophila, which will become the compound eye. So this is a high-resolution drawing of Drosophila imaginal disc, and you can see that there are these highly ordered cell populations called Ommatidia on the right. These are multiple cell types that are ordered in a certain way, spatially and functionally. In the middle, you have this blurry area, this very dense area called the furrow, and that's our wave that's going through from right to left. Actually, I think the image is inverted, so it's left to right. But the furrow is moving through this tissue. On the back side, you have the Ommatidia, the differentiated tissue. On the right side, you have undifferentiated cells that are sort of random and low density. And so the idea is as the wave sweeps through, these cascades become triggered, it contributes to differentiation, and you get this tissue that emerges. So this is an interesting model. The data is from C. elegans. The example is from Drosophila, but seeing that there are these mechanisms that are parallel are helpful in sort of understanding what proteins and genes are involved in this. The other paper we worked on in 2024 is this it's on hypergraphs in anatomical hypergraphs in development. So this is called Hypergraphs Demonstrate Anastomoses During Divergent Integration. We would like to add to it. I'll show you some of the work that might add on to this, might improve this work. But so basically we define hypergraphs as nodes and edges that are contained within hypernodes and hyperedges. So you can see that on the lower right you have these hypernodes, and inside each of these hypernodes you have a bunch of cells or nodes that are connected through edges. Those edges then can be bundled into hyperedges, and they can show sort of selective connectivity between structures. As we saw before, we have tissue types that have a bunch of cells in them. Those tissue types can be hypernodes. Hyperedges can be connections between different tissue types, whereas the nodes and edges can be connections between different cells, and they're maybe they're signaling relationships. So this can reveal subgraphs of selective connectivity. In anatomical terms, these are known as anastomoses, or connections between two ramified regions of the anatomy. We can see this in zebrafish on the lower left, where you get cardiac progenitor formation, some cell migration and fusion that results in a linear heart tube, and then chamber formation of the heart later. So you get this anatomical transformation process whereby the cells are being grouped into these tissues, and then they form these structures. And so this is an example of an embodied hypergraph based loosely on C. elegans embryogenesis. It's not exactly based on the lineage tree of C. elegans, but it's a demonstration. And so what you have is a single cell. You start with a single cell. You have doubling events, and so you end up with this gray set of hypernodes, which are sort of the undifferentiated cells. Very early on, the germline branches off and takes this yellow path. And then you have these other two paths that emerge from the undifferentiated cells at the 32 cell stage. So these are neural precursors and the embryo network of somatic cells, or what will become the somatic phenotype and the neural connectome, respectively. So the neural connectome is basically the cells that will become the nervous system or the connectome, and the orange cells will become the body outside the connectome. But each of these cells form a network in themselves, as we saw from the embryo networks earlier. And of course, we know that the connectome forms its own network. And the question is as these cell types differentiate, what are the connections between the connectome and the phenotype and the germline and everything else? Sometimes we expect really sparse connections. Sometimes things are totally modularized, like the germline. And sometimes you get cells that start off in one category and move to the other. So you might get cells that are somatic cells that turn into neurons through some process, not in C. elegans so much, but in other organisms. You get this interchange of cells from one category to another. So you see these connections between these subgraphs of the anatomy. Finally, I'd like to talk about Mehul's GSOC work. And of course, what he was doing was building on our hypergraph approach and trying to figure out how to decompose the C. elegans embryo spatiotemporally. So he built a number of hypergraphs using algorithms and other types of approaches. On the left, you see the lineage hypergraph, which is based on the lineage tree data. You have these concentric circles, which represent the area around single cells that allow you to sort of group different cells into these hypernodes. So you have a bunch of cells within each of these circles, and they're all connected in a network. That's based on the lineage tree. And then on the right, you have a spatial hypergraph where the relationships are based on the spatial distance between cells. So you can see it looks a lot different. The lineage hypergraph looks like a radiating tree. The spatial hypergraph looks like a bunch of clusters in different places. So that makes sense. You can also do sort of spatiotemporal decomposition using DB scan clustering techniques. And so he did this version where he applied this algorithmic approach and tried to find the nearest cells to, in other cells and, determine these clusters. And these clusters then served as the basis for these hypernodes. The hypernodes then being connected by multiple edges, which are collectively known as hyperedges, and you have your hypergraph. And so if we zoom in on this spatiotemporal decomposition, we can see that each of these cells have an identifier, that we might see in a C. elegans lineage tree. And we see clusters of these cells so that you get these hypernodes that are clustered, as well as the nodes themselves and their connections. So you can see that there's this fine-grained structure of this hypergraph. And so we haven't really gotten to the point where we can analyze the hypergraphs yet, but we can certainly build them from data in a number of sophisticated ways. So we zoom in even more and we can see the nomenclature, we can see their relationships, and these sort of the criterion that we're using, the concentric circles. So our weekly meetings continue this year. We usually meet on Mondays at 10 a.m. Eastern Time. It's in North America. And so you're welcome to join from anywhere in the world, provided it's not an inconvenient time for you. We also record our meetings and post them later on YouTube, so they're accessible asynchronously. And we do all sorts of things in these meetings. We do topical reviews. We talk about biology. We talk about computation. We do technological tutorials and other things as well. So we have an archive on our GitHub site where we have an archive of our meetings going back to the beginning of 2020. So there are a lot of topics in these meetings, a lot of content to catch up on. And certainly you can, I think, find a lot of interesting information in these meetings. So thank you to our contributors for October 2023 to November 2024. Bradley Alicea, Richard Gordon, Susan Crawford-Young, Tom Porteges, Jesse Parent, Paki Bonchalia, Mehul Aurora, Morgan Huff, Lucas Bastani, Hamanju Shogol, Vahid Gayumi, Hussein Ather, Harshit Sharma, Alexander Drysdale, Luke Noble, Sarav Kumar Roy, Daniel Paul, Jyothi Swarup, and Sushmant Reddy. So they at least attended one of our meetings. It probably did more, but I wanted to observe everyone who at least attended one meeting. And so, we'd like to grow our community, and you're welcome to join us, either attending a meeting or doing a project or whatever. And our topics are very wide ranging. So we've talked this year about things ranging from multiphysics modeling to tensegrity to biophysics to developmental connectomics to soft active materials, Janus phenomena, swarms and collective behavior, and solitons and differentiation waves. So thank you for your attention.